She's a professor in the Department of Zoology in Cambridge, and she's also director recently of the Cambridge Museum, which is an exciting new role she has. And so I'm sure she's had lots of experiences with opening a museum in COVID. And really excitingly, she was elected just last week to be a fellow of the Royal Society, which is a really big honor, and we want to congratulate her for that. I think her work is particularly interesting because it combines, its focus really is on social evolution. And this is not such a common focus. And she really looks at how this social evolution influences diversity. And then considers this both from a theoretical perspective, but also what important lessons we might learn for conservation. So I think this multiple approach she has to a really interesting question is quite fascinating. And she also works on multiple groups. So I learned first about her work on birds, perhaps logically, but then I heard her give a talk on beetles, and I was just amazed by how cool these beetles are. And so I guess today she'll be telling us a little bit about some aspects of her work, and it'll be super fun, I'm sure. And so with that, I'm really happy to have you, and I turn the floor to you.
Well, thanks very much. I'm just going to share my screen before we go any further. Okay, so is that working for everyone? And I can't see any reactions, so I guess it is. So thank you so much for inviting me to come and speak today. It's a huge honour to be here with you this morning. So this is a talk about the relationship between evolution and animal behaviour. And so it's a story that starts in the 1930s. And this is when the field of ethology, or the scientific study of animal behaviour in nature, really first started to take shape. And it was also a time of great expansion in evolutionary thinking. And as we shall see, these different fields of research generated quite different ways of analysing the connection between evolution and behaviour. So here are the fathers of ethology, who I'm sure you're all familiar with. It's Nico Tinbergen, Konrad Lorenz, and Carl von Frisch. And they were later jointly awarded a Nobel Prize in recognition of their efforts in establishing the field of ethology. And they look a bit grey and forbidding here, but their writings reveal just how much they were captivated by the vibrancy and richness and technicolor drama of animal behaviour in the natural world. And that's because it's impossible to look at actions like this, or even this, without being sucked into watching and wondering. And immediately all sorts of questions spring to mind. What on earth is going on here? Why are they behaving like that? And how are they even capable of doing something so amazing? Now, Darwin applied his theory of natural selection to explain the origin of behaviours like this, which he called instincts. And he noticed that within species, instinct varied from one individual to the next, and that some of this variation is inherited. And he suggested that natural selection accumulates variations which are useful, and that explains why closely allied species retain nearly the same instincts. Now, the ethologists built Darwin's ideas into their general framework for understanding any form of animal behaviour. And this is perhaps the most famous behaviour paper of all in ethology, which Tinbergen wrote as a 60th birthday present for Lorenz. And here he sets out 4 questions which we can ask of any animal's behaviour. And you can see that 2 of these questions, these 2 in bold here, follow in Darwin's footsteps by taking an explicitly evolutionary approach to just understanding the nature of animal behaviour. So they ask, what is the survival value of an animal's behaviour, or how is it adaptive or useful for the world in which it currently lives? And what is the evolutionary history of that behaviour? Is it shared by closely related species? And these are the evolutionary questions of a great naturalist, like Tinbergen, like Lorenz, and like Darwin himself, shown here in increasing order of beardiness. So they see behaviour happening in the here and now, And they ask, what use is that behaviour to the animal now? And how did it evolve to look like this? So they place the animal's actions at the end of an evolutionary sequence. Now, 5 years before Tinbergen's 4 Questions paper came out, George Simpson published his own questions about evolution and behaviour. And Simpson is famous for being one of the architects of the modern synthesis, the fertile period of evolutionary thought in the 1930s and 40s that blended genetical analyses of variation and inheritance with Darwinian ideas about natural selection. Now, Simpson was a paleontologist, and by this point, he'd already successfully introduced the concepts from the modern synthesis into paleontology, and now he turned his sights on doing the same for animal behaviour. Now, as a paleontologist, Simpson took a long view of evolution. And so that meant that he positioned behaviour in the middle of an evolutionary sequence rather than at the end, as the naturalists did. So he asks, how does behaviour evolve? But then, how does that behaviour determine the course of subsequent evolution? And it's this second part of the question, how does behaviour determine evolution, that I'm referring to as Simpson's question. So here we have 2 subtly different ways of thinking about the link between behaviour and evolution. Ethologists see behaviour as the endpoint of evolution, whereas Simpson thinks that any newly evolved behaviour will contribute to further evolutionary change. Now, as it turns out, Tinbergen's questions have dominated the way that we think about evolution and behaviour, while Simpson's question has largely been forgotten. And you can see that from this comparison of books from the Cambridge Zoology Library, because the volume over here on the left, holding Tinbergen's 4 question papers, is battered and bruised, and the pages of the paper itself are fraying and falling out because it's being read so intensively by generations of students and academics. But the volume bearing Simpson's question, by contrast, is pristine and immaculate and untouched. And yet, I think it is Simpson's question that matters to us now. So here we have a conventional description of the evolutionary process in which ecological conditions impose selection on variation that's inherited, which brings about trait change through genetic change. And Simpson's question asks, how does behaviour influence each of these steps in the evolutionary sequence? And that matters now because answering Simpson's question helps us to predict how animals will evolve and adapt in a changing world, and how their behaviour will help them do that. And of course, evolutionary biologists will have noticed that these links are also related to current hot button topics in evolutionary biology, which some people are now using to challenge the core concepts of the modern synthesis by developing a so-called extended synthesis. But as we can see, these are not extended questions, they're just neglected questions. They were formulated by Simpson, and so they're rooted directly, in fact, in the modern synthesis. So what I want to do in the talk today is to show you some work that we've been doing that addresses Simpson's question by investigating how behaviour determines evolution in each of these 3 different ways. Now, we can't possibly analyse the huge diversity of animal behaviour in our lab, so we focus on one form of behaviour in particular, and that's parental behaviour. And if we want to study how parental behaviour affects evolution, then ideally we want to study species that's locally abundant, that shows parental care, that breeds readily in the lab, and which has a sufficiently short generation time that we can carry out some experimental evolution so we can see exactly how behaviour can determine evolution. Unfortunately, precisely such an animal exists in the shape of the burying beetle shown here. Now, the natural history of the burying beetle was first made famous by the great French naturalist Jean-Henri Fabre. And here's the great man himself looking drained by the sheer magnificence of his own prose, because here is what he had to say about the burying beetle. The most vigorous and famous of these expurgators of the soil is the burying beetle, so different from the cadaveric mob in dress and habits, for he is a gravedigger, a sexton. He He buries a corpse entire on the spot in a cellar, where the thing duly ripened will form the diet of his larvae. He buries it in order to establish his progeny therein. He makes a clearance of death on behalf of life. And here, in much more prosaic terms, is the life cycle that Fabre was describing. So it starts here, and it's happening round about now, actually, as burying beetles come out from the winter and they fly around searching out the newly dead. And if they get lucky, then they find a carcass, a small dead body of a mouse or a bird. And if they get really lucky, then a partner will show up too. And then together, the pair of them will set about preparing this dead body for reproduction by converting it into a nest for their larvae. So they shave off the fur or feathers, they roll the flesh up into a ball, and they bury it below ground. And all the time they're doing this, they're covering it in fluids, in exudates from their mouth and from their anus. Now, while they're busy doing that, takes a couple of days, the female lays her eggs in the soil surrounding the carcass, and then the eggs hatch and the larvae crawl to the carcass, and there they take up residence in the nest and feed upon the flesh themselves. And the parents stay around too, and they both feed the larvae as well. And then within a week, there's absolutely nothing left of the dead body except for a few bones. And at that point, the parents fly off in search of new reproductive opportunities, and the larvae crawl away into the soil to pupate. And that takes 2 to 3 weeks. And then a week or so later, the adults that emerge attain sexual maturity, and the whole process starts again. So this is an animal that ticks all of our boxes. It's locally abundant in the woods surrounding Cambridge. It displays relatively elaborate parental care, and it has a short generation time of around 7 weeks, which makes it ideal for testing how behaviour determines evolution. Now, just before we get into the details of what we've been doing, I want to show you more about the general setup in our lab so that you can understand better the experiments that I'm going to tell you about. So this is one of our study sites near Cambridge. It's at Wycombe Fen. So we trap beetles here, and then we bring them back to the lab, and each beetle gets their own personal little box with a unique identity label on it and some soil for them to live in. And then when we want to breed them, we put them in a larger plastic box lined with soil, furnished with a dead mouse. We pop in a couple of beetles, we put the lid on, we put the box in a cupboard to simulate underground conditions, and then we come back a week later and we can measure the fitness consequences of that breeding attempt by counting larvae and weighing them. And this means that we can manipulate details of the environment during each breeding event. We can impose selection at each generation if we want to, and we can easily track the effects down the generation. And we can also process sufficiently large numbers of individuals that we can carry out some pretty large scale evolutionary experiments too. So we're going to start off by thinking about how behaviour changes ecological conditions. And we're going to do that by focusing on events that go on before hatching during carcass preparation. And this is work that was carried out by my star postdoc, Anna Duarte, and she was ably assisted by these illustrious gentlemen from the departments of genetics and biochemistry in Cambridge. Now, before we think about what the beetles do with the carcass, we should really see what happens to a mouse carcass that goes undetected by burying beetles in the hours after death. And it turns out that there's a surprisingly large number of videos on YouTube showing us precisely what happens. And so this is where I've stolen this video from. And what it's going to show us is time-lapse footage of a dead mouse in the hours immediately following death. So here we go. So here's the body, and the first thing you can see is that the abdomen is starting to distend. Mouse's gut are proliferating like mad, and the gases that they're producing are causing ballooning of the guts. So the abdomen gets larger and larger and larger, and eventually the pressure builds so high that the body wall ruptures. And at this point, fluids seep out, and this is an important step in the decomposition of a carcass because things really pick up at this point. And that's because rupture causes the rapid spread of microbes all over the dead body, and you can see that by the spread of these fungal hyphae. So what that means is that if a beetle wants to use the resources on a dead body to breed itself, it faces two major challenges. It's got to prevent rupture somehow, because that accelerates carcass decomposition and means that it can't use the resources on the carcass for its own reproduction. And it's got to somehow deal with the substantial competition that it faces from microbes that also want to colonize the carcass and use it for reproduction. So what we're going to do now is to look at how burying beetles rise to these two different challenges. And this time we're looking at footage that was shot in my lab, showing how beetles go about carcass preparation. And if you're at all squeamish, don't worry, it's all filmed under infrared light, so it's in black and white. And what we're looking at here is a pair of beetles making a carrion nest, and it's about 2 days' work condensed into 30 seconds. So the first thing you can see is that they move the body around a bit, but then they home in on the abdomen, they make an incision, and they pull out and they eat the guts. So that's how they deal with this problem of rupture, by immediately removing the guts. And then they shave off the fur. You can see this white stuff accumulating on the surface of the soil. That's the mouse's fur. And then they roll the flesh into a ball. And if we'd given them enough soil, the body would now be below ground. And so you can see that by the time we get to the end of carcass preparation, what we end up with is this beautiful sphere of flesh, glinting slightly in the light, because the beetles have covered it so comprehensively in exudates from their mouth and from their anus. So here's a close-up of the beetle's abdomen, so you can see what these lovely exudates look like. Here they are. And we've shown in several different ways that these exudates destroy bacterial cells and fungal cells, so they have antimicrobial properties. And we've also found that these antibacterial properties are only upregulated during reproduction. And you can see that from these data here, collected a while ago now by Sheena Cotter when she was a postdoc in my lab. So here we have the time course of reproduction. This is where we pair the beetles. And here we're looking at the antibacterial activity in the anal exudates. And you can see at the point of pairing, it's close to 0, but it rises through carcass preparation. It peaks as parents are provisioning their larvae, and it falls off thereafter. And Sheena went on to show experimentally that the reason why these antibacterial properties are only upregulated during reproduction is because the production of these antibacterials generates a fitness cost. So beetles simply can't afford to produce them all the time. Now, in more recent work, we've been interested in identifying some of the compounds in the anal exudates that confer antibacterial activity. And we exploited the fact that the antibacterial properties are only expressed during reproduction to identify which genes confer this antibacterial phenotype. So to do that, we compared the transcriptomes of guts from breeding and non-breeding females, and we focused on the guts because the exudates are extruded through the anus. And we decided to start by focusing on the lysozyme genes. And the results of that analysis are shown here. And as you can see, it turns out that burying beetles have 6 lysozyme genes. And for 5 of them, there's no difference in their expression in the gut, whether the female is breeding or not. You can see that relative expression levels overlap for these 5 genes in breeding and non-breeding conditions. But for this 6th gene, lysozyme 6, there's a whopping 15-fold increase in its expression during reproduction. So that strongly suggests that this lysozyme is an active component of the beetle's anal exudates. And Anna was able to provide further evidence to support this idea by carrying out qPCR analysis. And so the results of that analysis are shown here. So here we have the relative expression of the lysozyme 6 gene and the corresponding lytic activity of the anal exudates. And each of these data points here corresponds to a different female. And what you can see is a nice positive correlation between the two. And so it's rare evidence correlating individual variation in the expression of a single gene with variation in function. Now, obviously, there's lots of variation that's unaccounted for here. So clearly there are several other antibacterial agents in the exudates, and we're trying to work out what they might be in some of our ongoing experiments. Now, having discovered that these fluids have antibacterial properties, the next thing we wanted to find out was what they do to the bacterial community on the carcass. So to answer that question, Anna carried out the following experiment. And she had 3 different treatments, each replicated 6 times using soil from 6 of the different field locations where we trap burying beetles. So in the first treatment, Anna simply placed the mouse carcass on the soil, and then she came back after 24 hours and she sampled the bacterial community upon the mouse. In the second treatment, she did exactly the same, but this time at 24 hours, she added a pair of burying beetles, and they went ahead and they prepared the carcass for reproduction and they buried it as usual. And then when they'd done that, she removed the beetles and she sampled the carcass, the bacterial community on the carcass, so at 72 hours. Now in the third treatment, Anna played the part of the beetles herself. Now she didn't go so far as to apply any anal exudates, but she did bury the carcass, and again at 72 hours, she sampled the community, the bacterial community on the carcass. Now, the first thing that Anna did with these different bacterial samples was to measure the bacterial load on the carcass using qPCR. And what we expected to see was that the load would be much smaller on the beetle-treated carcasses than on either of the other 2 carcasses. But to our astonishment, that's not what we found at all. And you can see that from these data shown here. So here's our measure of bacterial load on the vertical axis, and here are the 3 treatments. The mouse sat on the soil for 24 hours, the beetle-prepared mouse, and the mouse buried by Anna. And you can see that to our astonishment, it's the beetle-prepared mouse that bears the greatest bacterial load, far greater than the bacterial load borne by mice in either of the other 2 treatments. So to try and understand why we found this puzzling result, we decided to look in more detail at the bacterial community on the carcass. And we did that by sequencing 16S ribosomal RNA genes. So here we have the results of that analysis, and it's just an example profile from one of our replicates. And there's quite a lot going on here, so I'm just going to walk you through it quite slowly. So these are the 3 different treatments. There's the mouse sat on the soil for 24 hours, the beetle-prepared mouse, and the mouse buried by Anna. And this is the bacterial community for the soil used in that replicate. And each of these different colours corresponds to a different class of bacteria, and the percentage reads that they represent in this particular sample. Now, the first thing you can see is that the act of burial introduces lots of gamma proteobacteria onto the carcass. That's shown by this grey box here. And this is also the class of bacteria that contains microbes that we know are pathogenic to the beetles. You can also see that they're very underrepresented on the beetle-prepared carcass. So clearly the beetle is eliminating these, presumably with its antibacterial anal fluids. And we call this process weeding. Now, the second thing you can see is that there are groups of bacteria on the carcass that's prepared by beetles that the beetle has added. They're things like Clostridia and Flavobacteria. And we think that these might derive originally from the dead mouse, from its gut, and from its skin. Now, remember that the beetle consumes the mouse's gut and associated microbiome during carcass preparation. And so what we think is that we're seeing some of these classes of bacteria from the mouse's gut passing through the beetle, back out through the anal exudates onto the surface of the carrion. And this is a process that we call replanting, because it's simply relocating bacteria from one bit of the carrion to the surface of the carrion nest. And then recent work by Danny Rosen's lab and Martin Kaltenpoth's lab suggests that some of the microbes on the carcass actually derive from the beetle itself. And they're gut microbes that the beetle carries that it inoculates the carcass with during carcass preparation. And this is a process that we call seeding. So these results in general explain why it is that the bacterial load on the carcass goes up rather than down. So yes, the beetles are weeding out some bacteria, but they're also adding a lot more on through this process of replanting and through seeding. And that explains why the bacterial load, we think, is greater when the beetles prepare the carcass than when the mouse is simply buried in the soil or sits on the soil. Well, the final step was to determine whether this replanting of the carcass bacterial community is adaptive from the beetle's perspective. And so to do that, we focused on the bacteria that were derived originally from the dead mouse and replanted on the carrion during carcass preparation by the beetles. And we did that by breeding beetles on dead germ-free mice. So we had two treatments. We had the germ-free mice and then a control mice with obviously germ-laden guts. And we bisected all of the mice. So we had a head end and a tail end, and that meant that we could isolate any effects of the gut microbes. And then we allowed the beetles to breed on these bits of mice in regular soil. And then at the end of reproduction, our key fitness correlate that we were interested in measuring was larval mass dispersal, because there's a huge amount of work has shown that this predicts both adult survival and adult fecundity. So the results of that experiment are shown here. Now over on the left here, we're just looking at the general relationship between larval density on the carcass, that's the number of larvae essentially, and larval mass dispersal. And this is for all the treatments pooled. And what you can see here is an unremarkable negative correlation, which simply tells us that the more larvae there are on this fixed pot of resource, the fewer resources each individual can attain, and so the lower the mass they attain by the time they disperse away from the carcass. And this is a very common result that we find always in our experiments, and it's not very surprising at all to see this trade-off between larval number and larval size. But it does mean that to isolate the effect of the carcass on larval mass, we have to control for this strong negative correlation. And so that's what we've done over here. And so once we do that, then you can start to see that there are some effects also of the carcass on larval mass. And you can see that the individuals that were raised on germ-free mice are generally lighter than their counterparts that were raised on the germ-laden control mice, and that the individuals that were raised on the germ-free gut end, the tail end, are particularly lighter than their counterparts on the control mice. So what this experiment suggests is that beetles are somehow adapted to the presence of microbes from the mouse, and particularly the mouse's gut microbes. And when we remove them, they perform less well. Now, we don't yet know what this adaptation is. It might be that the microbes are assisting the beetle directly with carcass digestion, for example, or it might be that their presence simply induces the parents to provide better care. But whatever the mechanism, the larvae are adapted to their presence on the carcass, so when they're removed, the beetle larvae perform less well. So what this body of work in total shows then is that beetles are changing ecological conditions by restructuring the bacterial community on the carcass, and that this in turn imposes selection back on the beetle by influencing the beetle's fitness. Now, what I want to do now is to move on and consider more direct ways in which behaviour can impose selection on others and influence the extent of genetic variation. And so the work I'm going to talk about now was begun by Ben Jarrett and Matt Schrader, and then continued by Darren Rebar in collaboration with Nathan Bailey and Sonia Pasquale. And so for this part of the talk, we're going to turn to events after hatching. So I thought I'd set the scene with some home movie of bearing beetle family life. So this here is the carrier nest. It was a mouse in a former life. And you can see snuggled up within are the larvae. And the larvae busy themselves by feeding away on the flesh of the carrion, all of them busy munching away. But then from time to time, a parent will show up. This is a female, and she seeks out individual larvae, and she makes contact with their mouth parts, and she transfers fluid directly into their mouths. And the males do this too. And it's because of remarkable behaviour like this, really, that the bearing beetles are often described as an insect that shows biparental care. But in fact, there's huge variation in the amount of post-hatching care that's supplied by parents from one family to the next. So some families, larvae are really lucky, and both parents hang around and care diligently like this for their offspring throughout the whole period of their development. But in other families, parents leave before their larvae have even hatched, and so larvae get no post-hatching care at all, and they have to fend for themselves on the carcass. So there's very broad variation in natural levels of parental care, and larvae can survive even without any parental care at all. Now, we exploited this quirk of burying beetle natural history to investigate how parental care is a source of natural selection. And so the design of our experiment is shown here, and it captures the kind of natural extremes in the supply of parental care. So what we did was we went out into the woods around Cambridge and we caught lots of individuals and we brought them back to the lab and we set some of these individuals aside. So they're our baseline population, and the rest we subjected to experimental evolution. So we bred them up to form an enormous founding population, and then we subdivided that population into 4 different populations with 2 treatments. And one of these treatments we called the full care treatment. So here we allowed the parents to stay with their larvae throughout the whole period of their development. And then we also had a no care treatment. And what happened here was that we allowed the parents to prepare the carcass. And once carcass preparation was complete, but before the larvae had hatched, we took both parents out. And so here the larvae had to fend for themselves. They got no post-hatching care. And we did that for generation after generation after generation. And then after 6 generations, we were beside ourselves with excitement, and we couldn't wait to see whether we could find any evidence that the populations had adapted to these different regimes of parental care. And so we tested for evidence of local adaptation. And we did that by putting all of the populations through a common garden environment. So all 4 populations experienced a generation of full care to eliminate any residual maternal effects. And then all populations either had full care or no care. So individuals drawn from the full care lines were exposed to both full care and no care, and individuals drawn from the no care lines were exposed to full care and no care. And we predicted that if each of these populations had adapted to its new regime of care, then we should see that the beetles from the full care populations should perform better in a full care environment than individuals from the no care populations, whereas individuals from the no care populations should perform better in a no care environment than individuals from the full care populations. And pleasingly, this time, that is exactly what we found. And you can see that from these data shown here. So the colour of the line reflects the line from which the beetles were drawn, so whether they came from the full care populations or from the no care populations. And here we have the care supplied to the current brood, either no care or full care. And a measure of breeding success, that's simply the proportion of broods where at least one larvae survived to disperse. And what you can see is that in the no care environment, individuals from the no care lines perform better than individuals from the full care lines, whereas in the full care environment, individuals from the full care lines outperform those from the no care lines. So it's clear evidence that there's been social selection by parents because individuals in the 2 types of populations have somehow adapted to these different regimes of parental care. And what we wanted to do next was to try and pinpoint the traits that had adapted. And obviously they could be in the parents, or they could be in the offspring, or they could even be in both. So we started off by looking at the no care parents. And what we found was that the main way in which they've adapted to having this imposition of a no care treatment is that they have accelerated how quickly they prepare the carcass for reproduction and how well they prepare the carcass. So that by the time we take them away, they've got much further on in the process than individuals from the other populations. And that's very well illustrated by the timing of the insertion of the final incision in the carcass. And this is one of the last acts of carcass preparation, and it involves the parents biting a small hole in the flesh so that larvae can more easily access the carcass, take up residence, and start feeding upon the flesh. And we've shown, as others have too, other labs have shown this too, that larvae have a better chance of surviving when there's a hole presence than when there's not. And what you can see from the data shown here is that the proportion of pairs that get round to biting the incision before hatching is far greater in the no care populations after 20 generations of experimental evolution than in either the baseline wild type or in the full care treatments. Now, it turns out that the larvae have also adapted to this different environment. And one trait that's changed in particular is the relative size of their mandibles. So that's these mouth parts shown here and dissected out, shown here. And they are key to larval survival without parents because they're crucial when larvae have to bite their own way into the carcass and start to feed on the flesh themselves. And so perhaps not surprisingly, it turns out they're now relatively larger in the no-care larvae than in the full-care larvae after 20 generations of experimental evolution. And you can see that from this plot here. So for any given larval mass, the larvae from the no-care populations have a larger size of mandible than the larvae from the full-care populations. And so that suggests that larvae have perhaps adapted to this no-care environment by becoming more self-sufficient. And then finally, we wondered whether larvae might also have adapted to having no parental care by evolving to help each other more instead. So to test this possibility, we ran the experiment shown here. So we started off by giving all the beetles in this experiment a standard carcass. So this was one that was prepared by beetles that had absolutely nothing to do with the evolving populations. They were stock beetles that we keep in the lab. And that meant that we standardized the environment on the carcass as much as possible across these different treatments. And then we created experimental broods of 10 larvae. Now, 5 of these larvae were our focal larvae, they're siblings, and they were either drawn from the full care populations or from the no care populations. And then we manipulated their sibling environment by giving them 5 siblings that were either drawn from the full care population or from the no care population. And so we had 4 treatments in all, where we have our focal full care larvae in a full care sibling environment or a no care sibling environment, and our focal no care larvae in a full care sibling environment and no care sibling environment. And then having set up these different treatments, we simply left the larvae to fend for themselves. And then we came back at dispersal to see how they'd got on. So there was no parental care supplied in any of these treatments, and that was intentional because we wanted to isolate the effects of larval interactions on larval fitness. Now, the way that we decided to analyse these data was to plot the mean mass of the focal larvae from each brood on the vertical axis, shown here, and the mean mass of the sibling larvae, making up the sibling environment in each brood, on the horizontal axis, shown here. So in this way, we could visualise the effects of the siblings on the focal larvae by looking at this vertical axis here, and we reasoned that if they attained a greater mass, that would be evidence of cooperation, whereas if they had a lighter mass, that would be evidence of competition. And likewise, we could look at the effect of the focals on the sibling larvae by looking at their mass along this axis on the horizontal axis here. And again, the greater the mass, the better the evidence for cooperation among larvae. And so here are the results of that analysis. And as you can see, they cluster by the 4 different treatments. So here we're looking at the full care focal individuals in a no care sibling environment. Here we're looking at the no care focals in a no care sibling environment. Here we have the no care focals in a full care sibling environment. And down here on the left, the full care focals in a full care sibling environment. And what you can see is that the larvae that perform best of all are when we have the no care focals in a no care sibling environment. And if we change the sibling environment and instead allow the no care focals to be raised alongside full care siblings, they do less well. The full care siblings cause them to attain a lower mass. The broods that do least well of all are the full care focals in a full care sibling environment, but we can promote the performance of the full care focals if we swap out that sibling environment and put them instead in a no care sibling environment, and then they attain a greater mass. So what that suggests to me, at least, is that the no care larvae are helping each other to attain weight, and so being cooperative, whereas the full care larvae are preventing each other from gaining weight and being competitive instead. So what we conclude then from this experiment is that when there is no parental care, then larvae evolve to be more cooperative. And without their parents to help them, the larvae compensate by evolving to help each other instead. And all this points to a general conclusion that parental care changes the actions of natural selection. And when we remove the supply of care, we expose offspring to a harsh new world with new selection pressures. And new adaptations for coping with this harsh new world then evolve in the larvae as a result. So what that means is that when parents stay to care for their young, as in the full care populations, they're really protecting their offspring from the harsh realities of this wider world. Or to put it another way, parents are relaxing the effects of natural selection on their offspring when they provide post-hatching care. And as we shall see next, that has knock-on implications for the extent of genetic variation that can build up and persist in populations. Now, to see how behaviour can influence the extent of genetic variation, we have to think about where genetic variation comes from. And of course, variation comes from genetic mutations of one sort or another that accumulate routinely over the generations. And the majority of these mutations have little or no effect on the phenotype of the offspring produced. So they're only mildly different from the wild type phenotype. But behaviour influences whether mutations are purged out of the population or whether they persist. And behaviour achieves that by influencing the actions of natural selection. So in the harsh environment experienced by the no-care populations, for example, the mutations are quickly selected against and removed because the abnormal phenotype is less good at surviving in these conditions. And by contrast, in the more benign environment created when parents are present, selection is relaxed and the mutation can persist. By this mechanism, you'd expect greater levels of genetic variation to build up and persist in the full care populations with parental care than in the populations with no parental care. So we tested this idea using the full care and no care evolving populations. And the logic behind the experiment is outlined here. So as we've seen, parental care allows mutations to persist. Mutations are likely to be mildly deleterious and probably recessive. And so that means we can only expose their existence by inbreeding. And we'd expect that the inbred lines should have lower fitness when they carry a high mutational load. And so our key prediction that we tested here is that after experimental evolution, inbred full care parents should have lower fitness than inbred no care parents because they're carrying the greater mutational load. So we tested this idea after 30 or more generations of experimental evolution. Took individuals from both the full care populations and from the no care populations, and then we inbred them for several generations and we measured the impact on their breeding success. And these are the results of the experiment. And what you can see is that as we predicted, the full cares that are inbred have lower breeding success than the no cares that are inbred. And this is consistent with the suggestion that they are carrying the greater mutational load. So what this experiment suggests to us is that by relaxing selection on their parents, parents are allowing mutations to persist, and in this way their behaviour is potentially maintaining genetic variation in the population. Without parental care though, these mutations are quickly purged, and so that's why when we inbreed the no cares, they have greater breeding success. And so in these different ways, behaviour influences the extent of genetic variation in the population. Okay, so now we've come to the end of the talk, and what I've shown you is 3 different answers to Simpson's question. We've seen that behaviour can influence ecological conditions and so impose and change selection acting on the individuals carrying out that behaviour. We've seen that behaviour can be a direct agent of natural selection, and we've seen that that has knock-on consequences for the extent of genetic variation that's maintained in the population. And more generally, what we've seen is that Darwin began analyses of the connection between behaviour and evolution, and that Tinbergen's questions, which incorporate Darwin's analyses, have shaped the way that we think about this problem for the last 50 years. But I wonder whether the time has now come for us to focus more on answering Simpson's question, because it shows us how behaviour can be integrated into the modern synthesis without the need for any extensions, and it speaks directly to the big problems of our time. And then just to wrap up, I'd like to conclude by thanking this absolutely fabulous team of people that have helped throughout all the work that I've described today. These institutions here, the Sanger supplied us with dead germ-free mice, and the Cambridgeshire Wildlife Trust allows us to collect beetles from their property. These fabulous organisations here have generously funded our work, so I'd like to thank them very much. And I'd like to thank you for listening so attentively. Thanks very much.